Moi drodzy, jak co roku na początku marca mam ogromną przyjemność zaprosić Was na nasze cykliczne wydarzenie Tydzień Mózgu, MariaMyśli na UMCS. Tegoroczne spotkania będą miały miejsce w tygodniu od 11 do 17 marca. Serdecznie Was na nie zapraszam. Jest to wydarzenie i szczególny rok dla nas wszystkich z dwóch powodów. W tym roku obchodzimy 80-lecie powstania naszego uniwersytetu i to wydarzenie wpisuje się w cały szereg uroczystych obchodów naszej rocznicy. Po drugie, wydarzenie jest przygotowane w ramach projektu W zdrowym ciele, zdrowy duch, kształtowanie postaw prozdrowotnych w ujęciu interdyscyplinarnym. Projekt jest finansowany przez ministra nauki i szkolnictwa wyższego w ramach programu Społeczna Odpowiedzialność Nauki. Koordynatorem całego projektu jest pani dr Aneta Unkiewicz-Winiarczyk z Wydziału Biologii i Biotechnologii, a Tygodniowi Mózgu swój czas poświęciła pani profesor Dorota Nieoczyn. Podczas tegorocznych obchodów Tygodnia Mózgu zapraszamy Was na interesujące wykłady, spotkania warsztatowe. To, co ja chciałabym szczególnie polecić, to spotkanie w unikatowej kapsule zmysłów, w których możecie poznać, zbadać swoje zmysły. Spotkanie jest organizowane z Instytutem Fizjologii i Patologii Słuchu w Warszawie. I najważniejsza sprawa. Mózgiem tego wydarzenia są nasi studenci. Ze mną jest Darina, prezeska Koła Naukowego Neurobiologów na Wydziale Biologii i Biotechnologii. Nasze koło współpracuje ze Studenckim Kołem Naukowym Psychologii ADESSE oraz Kołem Naukowym Kognitywistyki, również na naszym uniwersytecie. Darino.
Chciałabym zaprosić wszystkich chętnych do tegorocznego Tygodnia Mózgu. Zapraszamy.
Serdecznie Was zapraszamy.
Hi everyone and welcome to the Brain Week at UMCS. Today our guest is Professor Tiego Outeiro. Professor Tiego Outeiro graduated in biochemistry at the University of Porto and was an Erasmus student at the University of Leeds in the UK. Professor Outeiro did his PhD thesis at the Whitehead Institute for Biomedical Research, MIT, and worked at Fold RX Pharmaceuticals as a research scientist and consultant. Professor Outeiro was a postdoctoral research fellow in the Department of Neurology of the Massachusetts General Hospital, Harvard Medical School, where he focused on the study of neurodegenerative disorders such as Parkinson's and Alzheimer's disease. Professor Outeiro directed the Cell and Molecular Neuroscience Unit at IMM Lisbon from 2007 to 2014, and is currently full professor and the director of the Department of Experimental Neurodegeneration at the University Medical Center Göttingen in Germany. Professor Outeiro holds a joint professor position at Newcastle University in the UK and is invited professor at the University of Algarve. Professor Outeiro has authored more than 310 research articles in the international journals and participated in various international boards and in collaborative projects with the aim of identifying the molecular basis of neurodegenerative disorders, such as Alzheimer's and Parkinson's disease. He has been awarded multiple prizes and grants in Germany from the European Union and from other international funding agencies.
Witam wszystkich na dzisiejszym wykładzie. Naszym gościem jest profesor Tiego Outeiro. Profesor ukończył biochemię na Uniwersytecie w Porto i był studentem programu Erasmus na Uniwersytecie w Leeds, Wielkiej Brytanii. Profesor Outeiro obronił pracę doktorską Whitehead Institute for Biomedical Research, MIT i pracował w Fold RX Pharmaceuticals jako naukowiec i konsultant. Profesor Outeiro był stypendystą podoktoranckim na Wydziale Neurologii Massachusetts General Hospital, Harvard Medical School, gdzie koncentrował się na badaniach zaburzeń neurodegeneracyjnych, takich jak choroba Parkinsona i Alzheimera. Profesor Outeiro kierował oddziałem neurobiologii komórkowej i molekularnej w IMM w Lizbonie w latach 2007-2014, a obecnie jest profesorem zwyczajnym i dyrektorem Departamentu Neurodegeneracji Eksperymentalnej w Uniwersyteckim Centrum Medycznym w Göttingen, Niemczech. Profesor Outeiro zajmuje stanowisko wspólnego profesora na Uniwersytecie Newcastle w Wielkiej Brytanii i jest zaproszonym profesorem na Uniwersytecie Algebra. Profesor Outeiro jest autorem ponad 310 artykułów naukowych w międzynarodowych czasopismach i uczestniczy w różnych międzynarodowych radach i wspólnych projektach mających na celu identyfikację molekularnych podstaw zaburzeń neurodegeneracyjnych, takich jak choroba Alzheimera i Parkinsona. Otrzymał wiele nagród i grantów w Niemczech od Unii Europejskiej i innych międzynarodowych agencji finansujących. Zapraszamy wszystkich do przesłuchania wykładu oraz na kolejne prelekcje w ramach Tygodnia Mózgu.
Hello, everyone. My name is Tiego Outeiro, and I'm very happy to be here with all of you. I wish we could be together in person, but this is not possible, so it's still great to join you via Zoom. So what I'll talk to you about today is about unraveling the molecular basis of Parkinson's disease from biology to pathobiology. I'm a basic scientist, so that means I do a lot of laboratory research. I run a laboratory here in Germany where I'm based. And as an academic, as a professor, as a scientist, I've been very frustrated over the years because we do a lot of work, a lot of research that we are very excited about, but then it seems to be very difficult to translate what we find into clinical applications to make new medicines that doctors can use to treat their patients. So in academia, we talk about this valley of death. And as a scientist, this has been very intriguing. So I've been thinking about this for some time now, and I've been wondering what is the case, why this is the case. And if you look at all the data that exists for complex diseases like Alzheimer's disease, for example, and if you look at all the thousands of clinical trials that have been performed so far, this is quite scary because although we've tested many hypotheses in the different clinical trials, many of those hypotheses have been failing. So we've tested disease-modifying biologicals, disease-modifying small molecules, symptomatic reducing molecules, and a lot of them have failed. So fortunately, things seem to be changing. In the past couple of years, we've had 2 or 3 successes, although they are not great successes, but they give us hope that we might be on the right track. And if you think about Parkinson's disease, this is a disease described by James Parkinson more than 200 years ago. We know it's a movement disorder, and it's characterized by symptoms that include bradykinesia, and then patients may have rigidity and or resting tremor and or postural instability. So these are the typical motor symptoms of Parkinson's. But we need to define certain things. So let's look at first things first. So Parkinson's is a very complex problem. We typically think about this yellow part, that's the motor part of the disease. But we now know that in the premotor phase of the disease, so years before the onset of the motor problems, there's already other alterations taking place, such as sleep disturbances, constipation, hyposmia, depression. These are all typical in Parkinson's patients even before they show the motor symptoms. And what we know is that the success of the most famous therapy for Parkinson's, called levodopa, this has biased our view because it has pointed us only in this direction where we think about the movement problems that come because in the brains of patients, so in healthy people, when you look at the brains upon autopsy, if you look at this region called substantia nigra, you see that it's dark, it's pigmented by this dark pigment, and that means the neurons are there and functional. In Parkinson's, if you look at the same region, you see that those neurons are died, and so there's not as much pigment, the region is not as dark. And we also know that in Parkinson's, we find in the brains of patients these typical protein inclusions called Lewy bodies and Lewy neurites. This is typical when you look in an autopsy at the brains of people that died with this disease, then you find these inclusions in the brain. But we know that we need to pay attention to the premotor phase that is not related just to the death of those neurons. And as a scientist and someone that reads papers about this disease, this is a typical first sentence that you find in many of the publications in the field of Parkinson's. But we think this is not really complete, and we all perpetuate this because it's what we know and this is what we write about, but we think we need to change this. And today, we have a lot more biological information and also clinical information that we can use to do a better job at classifying Parkinson's disease. So what we have, in fact, is a problem of definition. We've been calling Parkinson's disease to something that can be very heterogeneous. And in fact, we should think about Parkinson's diseases and not just as one Parkinson's disease. So you can think of this as if it were a riddle, and this is something I just made up for illustrating the point. And so if something looks like it could be Parkinson's, it does not mean that it's the same type of Parkinson's that everyone else has. So we need to consider that there may be more than one Parkinson's disease. And when you think about the possible pathogenic mechanisms, when you study them in the lab, you see that the picture is very complicated. We have problems in mitochondrial function. You have pesticides that can induce Parkinsonian symptoms. You have problems in pathways in the cell that clean up proteins like autophagy and the ubiquitin proteasome system. You have problems in protein aggregation, as I told you about, the presence of Lewy bodies. You have neuroinflammation, and all of these seem to converge to lead to neurodegeneration, meaning cell death in the brains of the patients. So alpha-synuclein is an important component of this, and I will come back to this later. When we look at the brains of Parkinson's patients, neuropathology is not as simple as it seems, because we have also cases of people here named ILBD. This is a brain of someone that had ILBD, and this is a person that whose brain looks like a Parkinson's brain. So you see the typical alpha-synuclein inclusions, but this person never had any motor symptoms of the disease. On the other hand, you have here on the right an example of a patient that carried a LRRK2 mutation, a mutation in one of the genes associated with Parkinson's disease. And when neuropathologists looked at the brain, they were shocked to see that there was no alpha-synuclein accumulation. So alpha-synuclein does not always accumulate in the brains of patients with Parkinson's. But now, instead of requiring an autopsy to look at this, we can measure alpha-synuclein in cerebrospinal fluid. So there's an assay that was developed for prion diseases, other terrible neurodegenerative diseases, and now this has been applied to Parkinson's disease. And there was a recent study that confirmed the validity of this type of test in the field of Parkinson. So this was a very important study that came out last year, and that tells us that we can use this as a biomarker to distinguish people with alterations in alpha-synuclein from people that don't have those alterations, and we don't need to look in the brain. We can just look in biofluids that we can collect from patients. Another tool that is very useful is neuroimaging that measures neurodegeneration. So we can do and use this technique that enables you to image the brains of patients without having to wait for their death. And here what you see is a healthy control subject. So you see this red signal, this is normal. A Parkinson's subject has reduced signal in the basal ganglia, the region where you have the dopaminergic neurons. But you then also have these weird cases where someone has Parkinsonism but has normal signal in this type of imaging study. So neuroimaging is an important tool, but still it's not as clean as we would like. And then we know that we're all different. If you look around to your friends and colleagues, we all look different, and that's because genetics makes us different. But genetics explains only a minor fraction of neurodegenerative diseases. So we don't know why is it that as we age, some of us may develop this disease and others apparently not. Genetics is complex, as I told you. These are some of the genes, and this is just to show you that there's more than 90 variants, 90 genes that have been associated with increased risk for Parkinson's that we don't really know very well how they cause Parkinson's. But we can identify these genes because sequencing the genome these days, or sequencing genes, is very easy. So we can easily identify genetics. And then one could even consider additional layers of complexity that we don't understand very well at all. And this could have to do with the interplay between genes in the environment, and the mechanisms are called epigenetics. And I'm sure you heard about them. These include DNA methylation, chromatin modification, microRNAs or non-coding RNAs. These are all epigenetic mechanisms that can be influenced by the environment. So what is Parkinson's disease? It is everything together, and we need to integrate all this information from imaging, from genetics, from biomarkers, from clinical observation into something that hopefully then will enable us to distinguish and define Parkinson's disease. So I've been working with other colleagues from all over the world on the biological definition of Parkinson's. And we published also last year a first publication that discussed the need for defining Parkinson's disease better than what we've been doing today. And because we are now in a position to start defining and classifying Parkinson's better, there was an effort that was a sequence of the task force, and these are the colleagues that were involved. So we wrote a paper where we proposed a biological definition or biological classification of Parkinson's that was based on three components that I mentioned to you before: the synuclein status, whether it's pathological or not, the neurodegeneration that can be measured with neuroimaging, and the genetics that can be measured by looking at the genes directly. And of course, then the clinical observation by the doctors that see the Parkinson's patients is also very important. And we propose a classification system. You can then look at the paper. This is now published, and you can try to understand what it means. But basically, this is a matrix that we can use to distinguish different types of Parkinson's disease. And this is a very important tool for research at the moment, but it will eventually be very useful for clinicians as well in the future. But now let's look at the mechanisms. And we as scientists, we focus on very small parts of the big picture. So this is my fault as well in my research, but it's very difficult to look at the big picture and to understand everything that is going on. So as a scientist, I focus only on a small part of the problem, and we need to become better at integrating different types of information so that we can really understand the big picture. And this is illustrated here. So there's all this complexity, all these mechanisms, and what I'll do in the rest of my talk is focus just on one part that has to do with protein aggregation. This is something I've been studying for 25 years now, and we think this is still very exciting and important because we don't know how this connects to disease. We know this protein is very important in Parkinson's, and this is just a summary of some of the information we have. So we have information about the structure, about the function of the protein, the tissues where it exists, where the mutations are found. We have a lot of information, but we still don't really fully understand the function of this protein, especially in tissues outside of the brain, such as in red blood cells, where this protein is very abundant. And we think that maybe what can happen in different diseases where this protein can become pathological is that the protein may adopt different shapes. And these shapes can be influenced by the environment, by PTMs or post-translational modifications, by the cellular environment, whether it's more acidic, more basic, whether it has more metals or less metals, more energy or less energy. This may influence the shape, the conformation, the aggregation of this protein. And now there's also an evolution to this initial concept that maybe we had disease because proteins would just misfold, aggregate, and cause disease because they would become toxic. So this was the gain of toxic function mechanism. But we are now also paying attention to the flip side of this, which is if you have protein aggregates, then this means you have less functional protein. And if you have less functional protein, maybe you have proteinopenia, meaning you have less protein available to perform its normal function. And we are very interested in addressing this question. Does this protein become toxic? Does it cause synucleinopathies? And we know that aggregation is a continuum. So you start with soluble functional protein. The protein starts to come together and clump and form dimers, oligomers, protofibrils, and eventually it forms a large clump we can easily detect, and we call that a protein aggregate. But the question for us as scientists, when we look at a cell under the microscope, how do we see those tiny little species before they become an aggregate? And here, just to satisfy your curiosity, in case you haven't spotted him yet, that's where Wally is, so I can switch the slide now. But this is just to tell you that size matters. And when you look at the stars and at the sun, you know you need to use telescopes. When you want to use at the cell, you need to use microscopes. But when you want to look at proteins, you need to use even more powerful tools. And there's now super powerful microscopes that we call nanoscopes or electron microscopes that enable us to really see structures and proteins in great detail. And I show you here an exciting example of using these types of techniques. So here you have 4 different versions of this protein alpha-synuclein. You have the wild type, so the normal protein, and then 3 mutants. It doesn't matter what they are here. But what you can see is that when we look at cells with these types of powerful microscopes, you see that it looks very different. Like, for example, in this TP variant, we see inclusions that now we can distinguish as looking like rings. And before, we could not distinguish this because the microscopy tool we were using was not powerful enough. Another exciting microscopy technique we're using is called expansion microscopy. And this is very simple. This is based on the idea that you can use a matrix similar to that is present in the diapers of babies that absorbs the urine when they pee and keeps the water, keeps the liquid. It does not release it. And so the idea is that you can embed your sample in a matrix, in a gel, and do a series of treatments. These are complex treatments, but then eventually in the end, you can add water to your sample and the gel expands. And when the gel expands, it basically separates the molecules and the proteins so that now things that were too close together, you can distinguish now as different objects. And I show you here again some examples that are very exciting. So here are cells pre-expansion. So this is what we would normally see. You can see inclusions and the nuclei in blue. And then post-expansion, you can see that everything looks bigger. The nucleus looks bigger, the inclusions look much bigger. And when you zoom in with more detail, you can start to see those ring-like structures that I showed you also with the other technique, confirming that these are probably real, and we can now study what they mean for the cell. Now, when we think about alpha-synuclein, normally we think this is just a synaptic protein existing in neurons at the terminals, and maybe it's where it performs its function. But there has been this doubt over the years whether this protein exists also in the nucleus of cells. So we started looking at this, and we showed using mouse models, taking the brains and looking at the proteins in the brain and doing different treatments, we could detect protein in the nuclear fractions using biochemical fractionation. And the question was, is this something that happens only in mice, or does it happen in human brains as well? So it happens in humans. And here I show you human brain tissue, post-mortem, of course, where we use different antibodies to detect alpha-synuclein, and we could find signal inside the nucleus. So we were very happy to confirm that our observation in the mouse model was also real in human brain tissue, and we use different protocols. These are shown here, different staining approaches, different antibodies, and we always confirmed that we could detect protein in the nucleus. So this was important. But now what does this mean? If the protein is in the nucleus of the cells, what is it doing there? So together with our colleagues here in Göttingen, we use different techniques. This is nuclear magnetic resonance. So it's a biophysical technique to look at protein structure. And we basically put alpha-synuclein together with DNA, and we saw that there was a change in the signal. So it doesn't matter here what the spectra look like, but we basically saw that some of the amino acids changed their position in the context of the whole protein, suggesting that there was a change in the conformation. And why? So if the protein was interacting with DNA, what is it doing to DNA? And we found evidence that the protein is inducing DNA damage. So we used, again, a variety of techniques that enabled us to measure the integrity of DNA. And we could detect that there was damage. So here you look at these pictures, and these look like comets. So there's the head of the comet and the tail, and you can measure the tail size, and we could see that in cells expressing alpha-synuclein, we could detect an increase in the tail, meaning that there was more DNA damage. And we used other markers looking at histone phosphorylation, in this particular histone H2AX. This is a marker of DNA damage, and we could confirm that there was DNA damage taking place. And we did then transcriptomic analysis in cells and in mice, and we found certain genes that are related to DNA repair that were downregulated upon alpha-synuclein expression. So it seems that the protein somehow causes an effect in the cell that leads to reduced expression of DNA repair genes, meaning that maybe that's why there's more DNA damage. And again, that was done in models in the lab, but we confirmed this in human brain tissue as well. So this is looking in cortical tissue from patients with dementia with Lewy bodies. This is a disease that belongs to the synucleinopathy group, similar to Parkinson's, and we can measure an increase in this marker of DNA damage, suggesting that we could find double-strand breaks, curiously not single-strand breaks, but double-strand breaks in DNA, even in patients. So in summary, I told you a lot of information, and what I want you to take from this is that alpha-synuclein biology is complex. We still don't know everything that the protein does. We still don't know what are the toxic species and whether we can image them. This is something we're working on. I showed you that the protein can be found inside the nucleus, and we are wondering if this has any physiological or pathological function. I didn't show you, but we looked also at a post-translation modification called phosphorylation that seems to regulate the presence of synuclein in the nucleus. And now we're looking at whether the nuclear alpha-synuclein is pathological by asking whether it has seeding capabilities and whether it exists in different species, meaning different shapes. And we are also asking other questions such as, okay, if the protein is in the nucleus, is it also present, for example, in mitochondria? Does it interfere with energy production in mitochondria? We'll find out. So where are we in understanding and treating these types of neurodegenerative diseases? We have to think of them as a big iceberg. So in Parkinson's, we can identify the clinical symptoms that are related to the death of the dopaminergic neurons, and clinicians can treat the symptoms by using, for example, levodopa. But what we are still not very good at is at understanding the mechanisms and whether this has to do with protein aggregation or not. So we need to study the disease in the lab to understand the molecular mechanisms so that in the future we have therapies that enable us to treat those symptoms. And finally, I want to thank you for your attention. I want to show you some of the pictures in my lab that have worked with me over the years. A lot of collaborators. Science is great when we have the opportunity to collaborate and to travel and to visit the colleagues and discuss the science. So I hope the world will eventually become normal, that we can all travel and visit each other without problems. Yeah, I'll stop here. I'll show you other colleagues in Newcastle that contributed to this work. And thank you for your attention.
Thank you, Professor, for this really interesting lecture. We really appreciate you taking time to present for us today. And thanks everyone for joining us today and paying attention, and hope to see you on other